Okay, so today we're going to talk about hemoglobin. Hemoglobin makes up about 90% of an RBC's dry weight, which means your RBCs are pretty much just packed with these guys. And why is that? It's because the main function of hemoglobin is to bring oxygen throughout your body. And we will learn more about the specifics later on. But basically, long story short, is if you have really low levels of hemoglobin on your CBC, which is your complete blood count, you could risk dying. And this is because you're not oxygenated enough. That is how important hemoglobin is. So first of all, we must learn what it's made of. So what is it made of? Hemoglobin is made of 4 globin chains. It is a tetramer. There's 2 pairs of alpha chains and beta chains. And there's also 4 heme groups that has a protoporphyrin ring on it with a ferrous iron. Okay, ferrous iron. And what are protoporphyrins made? Protoporphyrins are made in the mitochondria, and it carries the ferrous iron. How are they made though? The main enzyme responsible in making protoporphyrins is the delta aminolevulinic acid, or DALA. And DALA is influenced by erythropoietin. And erythropoietin is made, I think, on your kidneys. It's a hormone that controls the production of RBCs. So now you see how it controls it. It controls the production of DALA, which controls the production of protoporphyrins. That's the big picture. So excessive porphyrins in the body is called porphyrias. And that's pretty much it. That's all you really need to know about protoporphyrins. So how about the globin chains? How are they made? So there's 2 chromosomes that are responsible for making globin chains. There's chromosome 16, which carries the 2 zeta genes and 4 alpha genes. And, okay, I forgot I had animation here. And chromosome 11 has 2 epsilon genes, 4 gamma genes, 2 delta genes, and 2 beta genes. So for a better way of putting it in a diagram, I made it into this. And so the direction of activation of these genes in order for them to produce different types of globin chains are from the zeta. For chromosome 16, it starts with the zeta gene and going down to the alpha, but there's only 2 in here, so that's really not hard to memorize. On chromosome 11 though, there's epsilon first and then gamma, and then gamma again, and then delta, and then beta. So think about it as like every time you see the word of the gene, it actually has 2 genes per word. So zeta has 2 genes, alpha has 2 genes here, and then another alpha which has 2 genes. So that makes it that alpha actually has 4 genes on chromosome 16. And then here it's the same thing. So epsilon has 2 genes, gamma has 2 here, and then gamma has 2 here. So that means gamma has 4 genes total. And then delta and beta with 2 genes each. So the first pair of chains, globin chains that are made during your embryonic development is actually a zeta chain and an epsilon chain. And then the next one will be an alpha and an epsilon. So see the direction of activation. It starts at the top and then the bottom. And then another one from zeta with gamma this time. And so these are the first hemoglobins that are produced in the body. These are the embryonic hemoglobins, which is the Gower 1, Gower 2, and Portland. They're pretty much like non-functional hemoglobins, so you pretty much get rid of them before you're born. So during fetal development, after the first 4 months of development of a baby, hemoglobin F gets produced. And hemoglobin F is a pair between alpha and gamma. So this is the final percentage of how much hemoglobin F is expected in a normal adult. And in adult hemoglobin, you have hemoglobin A2 as well and hemoglobin A. So as you can see, after birth, you only have alpha on chromosome 16 that's still activated. And alpha creates hemoglobin F with gamma. And then alpha with delta creates hemoglobin A2, and alpha with beta creates hemoglobin A. Hemoglobin A is pretty much our standard hemoglobin. When we say hemoglobin, that is what we're trying to say. The one with 2 alpha chains and 2 beta chains. And this is our normal hemoglobin. And so this slide is really helpful because you're able to see how much of each you have in adult hemoglobin. And these are also helpful because you can see the development of different types of hemoglobin throughout the embryonic development and the fetal development as well. So now, the next topic would be the iron accumulation. Why would I be talking about iron accumulation? Well, we're talking about hemoglobin, and well, pretty much it's because if there's an impairment in the production of either the globin chains or the protoporphyrin ring, it doesn't really affect the amount of iron that your RBCs are going to accumulate. Globin production is directly related to porphyrin production and vice versa, which means protoporphyrin production is reduced when globin production is impaired. However, iron continues to accumulate in RBCs regardless of these conditions. So what I'm trying to say is, iron still is present on your RBCs regardless of whether you are not producing enough globins or porphyrins. And they would be seen on your RBCs if you stain them with Prussian blue stain. So sideroblasts are iron-laden nucleated RBCs. Siderocytes are iron-laden anucleated RBCs. Ringed sideroblasts are RBCs with an accumulation of iron around the nucleated RBC. So they look like this. You see the purple things, those are the iron accumulation. It's kind of how you would see it on a microscope, so I'm just like doing my own diagrams here. And hopefully that helps you visualize what it would look like on a microscope, but it kind of looks like this. Remember, you wouldn't see this on a right Gemsa stain, you would only see this on a Prussian blue stain. Prussian blue is the stain for iron. And that's pretty much it for today. So these are the things that I want you to take away from my presentation. So remember that hemoglobin is made of the tetramer, alpha globins, and 2 beta globin chains with 4 heme groups. Remember that DALA, delta aminolevulinic acid, is the one responsible for the production of protoporphyrins and is influenced by erythropoietin. And that is how it controls the production of RBCs. Remember the different types of hemoglobins, the embryonic, fetal, and adult, and how much of it is present in adults. So basically, you just want to know, what's abnormal and what's not. And lastly, iron accumulation. And that's pretty much it. Thank you for taking your time to learn today, and I hope this helps. Bye!